The major differences between the bacteria, Eubacteria and Archaebacteria are: so in Eubacteria, the nucleus is absent, whereas and also in the Archaebacteria, nucleus is absent. So nucleus is absent in both of them. So we are comparing the Eubacteria and the Archaebacteria here. And if we talk about the unit membrane bound organelles, then they are absent in both of these bacteria. Cell wall consists of peptidoglycan in case of the Eubacteria, whereas in case of the Archaebacteria, there are variety of types. There is no peptidoglycan present in the cell wall. Instead, the Archaebacteria consist of the pseudomurine in their cell wall. So they lack the peptidoglycan, whereas the cell wall of the Eubacteria consists of the peptidoglycan. So in case of the Archaebacteria, they are having pseudomurine, which is a repeating disaccharide unit. Now, if we talk about the membrane lipids, so they have the ester-linked straight chain fatty acids, and in the Archaebacteria, they have the ether-linked branched aliphatic chains. The transfer RNA in case of the Eubacteria is thymine. Thymine is present and formal methionine carried by the initiator transfer RNA. And in the Archaebacteria, no thymine is present and methionine is carried by the initiator transfer RNA. So in the Eubacteria, the ribosomes, in case of the ribosomes, the elongation factor 2 does not react with the diphtheria toxin, whereas in the Archaebacteria, it reacts with the diphtheria toxin. Now, the sensitivity to the chloramphenicol, which is an antibiotic, so the Eubacteria are sensitive to it, but the Archaebacteria are insensitive to this. The sensitivity towards the anisomycin, so they are insensitive, whereas the Archaebacteria are sensitive. Now, the DNA-dependent RNA polymerase, number of enzymes present in the Eubacteria are 1, whereas in the Archaebacteria there are several number of enzymes which are present. Rifampicin sensitivity, if we talk about it, it is a drug, it is a antibiotic. So in case of the Eubacteria, they are sensitive towards this, whereas the Archaebacteria are insensitive towards the rifampicin. And the polymerase 2 type of promoters is absent in the Eubacteria, but it is present in the Archaebacteria. Metabolism, so methanogenesis is absent in the Eubacteria, but it is present in the Archaebacteria. So if we talk about the methanogens, so Archaebacteria are methanogens. Methanogens are the obligate anaerobes, and they can produce the methane as the end product by combining the hydrogen with the carbon. And there are no other bacterial methanogens, only the Archaebacteria are the methanogens. So chlorophyll based photosynthesis is present in case of the Eubacteria, but it is absent in case of the Archaebacteria. सो अगर हम देखें कि उन दोनों के अंदर सिमिलैरिटीज़ क्या हैं और डिफरेंसेस एक बारी पूरा फ्लो चार्ट में देखें, सो बोथ ऑफ दीज बैक्टीरियाज आर prokaryotic, both of them has no nucleus, both of them are single cell, both have ribosomes. Both of these bacteria do not have the membrane bound organelles. But what is different in the Eubacteria? Eubacteria's cell wall consisting of the peptidoglycan, whereas the Archaebacteria does not have peptidoglycan in their cell wall. Eubacteria cannot survive in the extreme environment, whereas the Archaebacteria can survive in the they live in the extreme environments. In fact, we can divide the Archaebacteria into the several categories, and they are also known as the extremophiles. Why they are known as the extremophiles? Because they are found under the extreme environmental conditions. Therefore, Archaebacteria are known as the extremophiles. Like they are the thermophiles. Thermophiles means the bacteria which are heat-loving bacteria, which live in the hot springs, which live in near the hydrothermal vents. and they can survive in the temperature up to 110 degrees Celsius. And they're also extreme halophiles. Halophile means that they can survive in very extreme high salt concentration also. So the archaebacteria are the extremophiles. They can live in the extreme environments. In fact, they live in the extreme environments, right? They are known to live in the extreme environments. And the bacterias, eubacteria, can be divided into the class bacteria and the cyanobacteria, whereas archaebacteria can be divided into the thermophiles, halophiles, and methanogens. So if we talk about the thermophiles are the heat-loving archaebacteria which are found in the hot springs, and the optimum growth temperature for them is 70 to 110 degrees Celsius. And most of them are sulfate metabolizers, and they may be autotrophic or they may be heterotrophic. So if we talk about the next class, which is halophiles, so halophiles means that they can live in the extreme high salt concentration and high saline concentration, high salinity habitats, like for example the halobacterium, right? So next we have the methanogens. Methanogens, as I told you, methanogens are the obligate anaerobes, and they can produce methane as an end product by combining the hydrogen with the carbon. And methanogens means the bacteria which can produce the methane. So that was the comparison between the eubacteria and the archaebacteria. So that's all for today. If you like the video, please share it with your friends too, and don't forget to subscribe the channel for more updates. Thank you and have a wonderful day.